Hey everyone, for lab this week we are going to be quantifying the metabolic rates of a range of animals. And when you get to lab this week, what you're going to encounter is a box full of supplies that looks something like this. And part of your project is going to be to assemble these supplies into a metabolic chamber and assembly that will allow you to measure metabolic rates. And in a nutshell, what we're going to be doing is we're going to flow air through a metabolic chamber, and there's a range of chamber sizes that you'll have available, but here's an example chamber. And we're going to flow room air where we know what the composition of the air is. We'll put an animal into the chamber. Then as air comes out of the chamber, we'll then use an analyzer to quantify the oxygen concentration of the air that leaves the chamber. And if we know the partial pressure or concentration of oxygen and air going in, and we quantify the partial pressure or concentration of oxygen coming out, then we can use that to calculate how much oxygen an animal is consuming. But one of the biggest challenges with doing this is just kind of getting our heads wrapped around sort of the setup that's involved in measuring an animal's respiration rate. So what I'm going to do first is I'm just going to walk you through some of the pieces that are in this box, and then I'm going to walk you through kind of the thinking for how all the pieces are assembled to each other. And then we'll look at an example assembly and some data collection. So the first piece in this box to pull out is this is actually not going to use the Power Lab unit. Instead, we're going to use an equivalent transducer published or made by Vernier. So this is the Vernier Lab Pro unit. So that's going to be kind of our central data collection unit, and that will interface with the Max just using a USB cord. So we've got that. Along with that, you'll find a gas pump in your kit. So this gas pump is what's going to flow gas through our chamber. And with the gas pump, what you're going to want to do is there's a piece of very long tubing on your table. You want to collect that connect that long tubing to the back of the of this gas pump. And all these connections between these different pieces, this is called a lure lock system, very similar to what's used for syringes. So these pieces will just twist together to connect. So that's going to be your gas pump. So that's going to flow the gas through. Now for the other end of your tubing, one thing that really helps us get stable measurements is if we work with air that's relatively uniform in consistency. And a cheater method for doing that is actually to take just a giant large carboy and stick your tubing into the bottom of the carboy. So you're just sampling air out of the carboy instead of just air that's in the room. Because the air in the room, we're breathing it, it's moving around, its composition is a little more variable. So then after we've got our gas pump connected in, and if you would like, this ring stand here has some attachment points to kind of keep everything organized. So we're going to pump air through this gas pump, and we'll need to connect that up to the power supply in a little bit. From there, the next thing we're going to do is we want to remove water that's in this air, because the relative humidity can have a dramatic impact on the precision of the oxygen measurements that we get. So to scrub out the water that's in the air, we also have this set of scrubbing columns. And the scrubbing column is filled with a substance that's referred to as dryrite that is just very good at bonding with the water that's in the air. And this dryrite is blue when it is dehydrated, and it will turn pink as it picks up water from the air. So then what you'll notice is once again, we've got a female connector here on this gas pump, and then there's a male connector end here on the scrubber column, and so you can just twist those together, and that's got some Velcro on it to hold it in place. So we're going to then start scrubbing the water out of the air. I'm actually going to lower this stand a little bit. Okay, and then the next thing we need to do is we need to work with air that is moving through the chamber at a known rate. We need a known rate so that we can then calculate the volume of air per unit time. So the next piece in our assembly is going to be a little flow controller and flow meter. So what you'll notice in this when you look at it up close is that it's got a little metal ball inside of here, and we've also got a little gauge here to tell us our flow rates. And then there's a little twist adjuster valve here that will let us adjust the flow of air traveling through this little flow meter. So this is then going to connect up. So again, we've got a female end here that will twist and connect into the male end that will twist and connect into the female end on our scrubber column. And then we've got a loose female end that's going to connect with the next part of our assembly. So once we've got air at a known rate, it's been scrubbed so it doesn't have any water in it, then we're ready to actually put our chamber in line. And in your kits, you're going to find you have a set of chambers that vary in size. So I just showed you the largest chamber at first. Here's a medium-sized chamber. You should also have a small chamber that looks like this. And then I actually created for us a set of micro chambers. This is actually just made out of a 5cc syringe. But syringes are actually really great metabolic chambers for measuring respiration rates for smaller animals. For the sake of this setup process, I'll just work with this medium-sized chamber. So for the medium-sized chamber, again, you should see it should have a male and a female end. This one actually has two female ends, so I bet there's another chamber somewhere in the lab that has two male ends to it. So we'll get that straightened out in a little bit. So what you're going to want to do, so for the sake of my demonstration, I'll instead switch to the small chamber. So then I'm going to twist in the female end from my flow controller into the male end of the chamber, and then I've got the chamber set and ready to go. Let's point this down a little lower. Okay, there we go. All right, so there's my chamber. Now, when we're ready to measure respiration rate, I can put an animal into this chamber. Both ends come off. One thing that we're going to need to do when we actually prepare this is we're going to need to make this chamber airtight. So you should also have some sticky tack in your kit to just close off the ends of the chambers really nicely. As I've been trying to emphasize in lecture, an animal's metabolic rate, particularly for an ectotherm, is going to be very much dependent on the ambient or environmental temperature. So it's also important while we're doing these projects to measure the temperature in the metabolic chamber. So there's a set of thermocouple probes that will actually fit into an opening in these chambers so that we can then get the temperature inside of the chamber. That temperature probe is going to connect into the vernier unit. So here I'm just going to connect it into channel 2, and I'll just click in place like that. Okay, so now we've got, let's say we've got an animal in here that is breathing, and so it's producing carbon dioxide, and it is also going to release water, and it's going to be drawing down the oxygen content in the chamber. The water that it produces, metabolic water, can also cause problems down the road. So the next thing that we're going to put in line with our chamber is actually a little condenser unit that looks like this. And this condenser unit, we're then going to put into a beaker that is full of ice. So that's going to cool down the air coming out of the chamber, and it will cause any water that's in the air exiting the chamber to condense inside of this cooling unit. So that's just going to get connected in line again. And it's going to look a little wacky right here, but in general what you want to do is figure out a way to get your animal chamber kind of stabilized in place, so it's not sitting at a jaunty angle. Okay, so now we've got air that has exited the chamber, has had the water scrubbed out of it, again, or condensed out of it using this condenser unit. So then the last piece in line for us is going to be a little oxygen sensor unit. So this oxygen sensor, it's a little fuel cell sensor. And you'll notice again it has tubing running through it. So we're just position that up on this bar. And then I'll connect in the female end here. And then the air can finally exit the whole system out of this female end as well. And I'm just going to position this up here as well, just to keep it out of the way, kind of keep everything organized. All right, so that's our plumbing setup. So just to review that one more time, and note that you could substitute in the micro chamber instead of the small chamber or the medium-sized chamber or the large chamber in this whole system. And there's also one other piece that you'll find in your box, and we'll talk about this a little more once we're in lab. This is actually a flow restrictor unit, so this little blue guy here. And this will be very useful for measuring respiration rates in very small animals. Okay, so I've got this whole setup put together. So then, in addition to connecting in my temperature probe, I also need to connect in my oxygen sensor. I'll do that right here. And this also, as with our Power Lab units, it's got inputs for channels 1 through 4. So I've got that entered in, and then I can connect that up to the computer. All right, so these Vernier units, actually they use a printer style USB cable. So I'm going to plug in the square printer end onto the little USB symbol on the Vernier Lab Pro transducer, and then on the Mac, I'll just plug this USB cable into the back of the Mac. Okay, so now once you've got your respirometry setup all put together, you've got your Vernier plugged in and interfaced with the computer at the USB, what you want to look for is a piece of software called Logger Pro. So Lab Pro and Logger Pro go together. That's this little icon here with a little set of calipers to it, and you'll click that to boot that up. Okay, so as with a lot of our instrumentation, one of the first things we're going to need to do is we're going to need to calibrate our analyzer. So what we're going to do is we're going to go to the channel 3 or experiment drop-down menu here. Then we're going to go down to set up sensors. And in the set up sensors, we're going to select Lab Probe number 1. Okay, this is going to bring up a little Lab Pro setup menu here. And in this case, I've got the oxygen analyzer plugged into channel 3. So I'm going to come over here and I'm going to select channel 3. And I'm going to choose sensor. And in this case, the sensor I'm going to choose is oxygen gas, right? Since I'm using an oxygen sensor in this case. All right, so now you may also have a flow meter depending on the setup. If you do, plug that into another channel, and you'll also want to set that up in another channel. And then in this case, in channel 2, the sensor that I have is a thermocup or a temperature sensor, and this is actually a stainless steel temperature sensor. Okay, I still have a couple details to work out with the temperature sensors. We may also need to calibrate those, and I'll have updates for you at the start of lab for the temperature sensor. For oxygen, what we're going to want to do is we're going to want to calibrate with gas that has no oxygen in it, and then we'll calibrate with room air as well. So for the no oxygen gas, what I've done is I've gone and I've filled up our gas bag with nitrogen gas. So what we can do for this part of things is we're going to connect this nitrogen gas up. Make sure that the clip is open so gas can flow. We're going to just clip or connect that into our gas pump right here. Okay? So I'll connect that in. And then to get the gas to actually flow, we do need to plug in our pump. Plug in this pump. And as soon as I plug that in, gas will be flowing. Just as a general note, try to avoid running the pump if you're not actually measuring something, because if you continually run it, you'll just wind up burning all the way through your scrubber, and then you won't be able to scrub out water any longer. So now I've got gas flowing from this. The other thing that I can see at this point is in my flow meter. I can't actually see the flow moving. And this is moving at a rate of close to between 0.9 and 1 liters per minute at the moment. The flow rate's not too important at this point in time. We can just let it flow with what it's got. Okay, so now if I want to calibrate, I can just click this oxygen sensor icon, and there's a little calibrate menu up top. And that's going to open up a dialog that looks like this. And what we're going to do is press the Calibrate Now button. And we're going to let this run for a little while. So it's giving me a voltage reading. And generally you want to wait for about 2 minutes until the voltage reading starts to stabilize, right? So right now it's slowly going down. It's gone down to 0.39 volts, 0.38. There's a fairly long lag time for getting a stable reading. So, and part of that is because this nitrogen gas has to flow all the way through this whole setup, all the way to the oxygen sensor at the very end of the line, right? So it's going to take a little while to get a stable voltage reading. You want to really wait for a couple of minutes for that voltage reading to get really close to zero, or at least to a very stable measurement, okay? All right, so for the sake of instruction, I'm going to just say that now let's say we've reached that low stable reading. And at this point, since we're just flowing nitrogen gas through, we're working with 0% oxygen. So where this says reading number 1, I'm just going to type in 0. And then I'm going to click this keep button. All right. Then what I can do is I'm going to unplug my oxygen or my nitrogen bag, and then I'm going to plug in the room air tubing, okay? Maybe I'll hang this back up again so it doesn't make as much noise. Okay, and what you should see is now this voltage reading is starting to climb up. So again, I'm going to just wait for a little while until that voltage reading starts to stabilize. And then you're going to want to enter in the concentration of oxygen in the air as your second reading, right? So that's going to be 20.95, or in this case, we can just enter in 20.9. So I'm going to type in 20.9. Okay, that's looking stable, so then I can press keep. Okay, and then my oxygen analyzer is calibrated. So I can close out of that window. Okay, now what you're going to want to do is collect some data with an empty chamber. So this is going to give us our inflow oxygen concentration. All you need to do in this menu is click this start button. And it's going to give you a little spreadsheet readout of oxygen, and also in this case our spurious temperature measurements. And what you want to look for is a relatively level measurement here, okay? If it's relatively level, all you need is about a 2-minute time frame, okay? And here, what you can do is, once you've got the data that you need, very much like with chart, you can select a data range. If you go up to the Analyze menu, in this case, and then go down to Statistics, that's going to give you the mean within that range that you just selected. Okay, record that in a spreadsheet somewhere, all right? Okay, so once you've got your baseline data, then the next thing you're going to want to do is put your animal in the chamber, continue collecting data. And so what I'm going to do here is I'm just going to start collecting data, and rather than putting an animal in the chamber, what I'm going to do in this case is I'm going to breathe into this chamber a couple of times, just as kind of a sanity check to see, is this going to give me a reliable measurement? This is actually a very common method for checking to see if your chamber and everything is behaving as you'd expect it to. And like they say, don't try that at home in a pandemic. Okay, so there's a good thing to learn from this right now, which is there's been a very tiny dip in the oxygen concentration from my initial breathing practice. What that indicates is most likely my flow rate right now is so high that I'm not going to be able to pick up changes in oxygen concentration very sensitively. So what I'm going to do now is I'm going to go back over to my flow meter, and I'm going to start dialing down my flow rate. So in this case, I'm going to aim for a flow rate that's closer to about 400 mils per minute. Okay, then I'm going to repeat my breathing effort. So I'm continuing to flow gas through the oxygen analyzer. And now we can see a more substantial drop in the oxygen concentration. Now here's the thing to realize. The amount of oxygen that an animal is going to consume out of that 20.95% is relatively small. So right here, this axis is only really dropping down to about 20.79 at the very lowest, and now it's starting to come back up again. And so when you actually have an animal in the chamber, what you should observe is you should see a period where the oxygen concentration goes down. And then what you want to look for is, once again, kind of a relatively flat and stable portion of your reading. And you just want to do the same thing. Analyze statistics and calculate the mean. Since this is still running, it's going to wait until I stop. So now I could analyze statistics, and there's the mean for that section of my recording. In the event that the temperature probes misbehave, we can also record temperature just using one of the BAT thermocouple probes instead. Alright, so in the lab handout, what I want you to do over the course of lab is measure metabolic rates in at least 3 different animals, ideally at approximately the same temperature, generally around room temperature. And then towards the end of the lab, there's 2 different exercises to characterize metabolic rates in 2 different contexts. One is to examine how metabolic rates change as group size changes. So for that, basically, what you want to do is you can do this with crickets, or potentially with one of the other animals that we have. Start out with a smaller number of individuals, compare that to a larger number of individuals. The other option will be to explore on temperature responses on how metabolic rate is influenced by changes in temperature. So read more about that in the lab assignment. But I hope that you'll find that measuring animal metabolic rates, there's a little bit involved to the whole setup, but once we get running, it can be pretty fun.